So what I'd like to talk about today is a project that we did looking at the plants of Florida. And yeah, great. If you could go to the first slide. Thanks, Israel. So Florida, a lot of people think of Florida in terms of Disneyland and Miami, but Florida, of course, itself is a really diverse set of ecosystems and habitats. And there are about 4,100 species of plants that reside in Florida, making it, I believe, second only to California in terms of plant diversity. And just to give you a little bit of background on Florida plants, if you look at the northern area, so I'm just going to talk about three really general regions of Florida, but Florida, of course, if you zoom in, you will see a lot more mix of mosaic and habitats that can be found there. But if you look in the north, you often see this really like oak scrubby area where you have kind of a mixture of lots of diversity of plants that are able to kind of withstand a little bit colder temperatures in the winter, and a lot of plants that extend their ranges into the rest of the South. If you go into central Florida, there you will see where we often see a real mix of northern plants and southern plants kind of coming together. But right in the center of the state, you can see this area that's a really like scrubby, sandy habitat shown by this second picture. And this is a picture. There's an area in the middle of Florida called the Lake Wells Ridge. And this area is thought to have been separated from the mainland during, glacial periods in the past when sea level rose and causing, there to be some sort of insular speciation happening there in this area to be separated. So you have this really scrubby habitat there in the middle. And then of course, when you go down into South Florida, we have this really marshy area that we know of with the Everglades, and a lot of plants that are really adapted to this marshland. So these are three really broad regions, but of course there are, as I said, a lot more diverse, habitats. If you zoom into any one of these pieces, each part of Florida feels like you're moving through a different habitat. And the anthropogenic impacts on Florida have certainly been large, with large scale agriculture happening, and of course massive development, with certainly along the south east coast, where you've got Miami all the way up to West Palm, where the whole area is really just a bunch of skyscrapers and large cities. So the conservation priorities for Florida are really quite high in terms of trying to understand how these anthropogenic effects are certainly impacting these areas, and what do we know about these areas from a spatial phylogenetic standpoint. So if you'll go to the next slide, Israel. Thank you. So these are the three main points that I talked about here. And I just kind of want to point out that these have been defined into three main ecoregions, right? So you have this kind of northern one shown here at the top, this really diverse sort of central one here that has this sort of scrubby habitat. And then of course the Everglades is kind of its own unique ecoregions. And I just want to highlight that so we can kind of, it's fine, you can go to the next slide, so we can sort of pinpoint that as we look at some of these results coming in later. So one of the questions we wanted to ask is, what do these communities look like from a spatial phylogenetic perspective? And two sub questions under that are, do we see patterns that highlight these ecoregions? So can we detect these ecoregions with spatial phylogenetics? And are areas of conservation concern from a spatial phylogenetic standpoint being protected? All right, so to do this study, we did very much what we've all kind of been talking about, of course, is get the spatial data and the phylogenetic data. For the spatial data, we took advantage of all the amazing digitization efforts that have been happening in museum collections and collected data from a number of herbaria found in the Southeast. And then in terms of the points that we collected points across these entire ecoregions, so not just points in Florida, with the idea that we're trying to really collect the entire niche space of each of these species, so we know the entire temperature range each species can handle, for example. And then we limited this study to plants that are only found in Florida. And then, of course, we built a phylogenetic tree. This tree was built using molecular data that was found online, and also that we sequenced using museum records as well. or museum samples. And so on the if you'll just go back up one slide, thank you. So on the left side here is the survival time, right? So this is the chronogram. This is the time dated phylogeny, where we can kind of examine communities in terms of time that they've been together. And on the right is the phylogram, right, showing character divergence. So I just kind of like split it up this way so you can kind of see the differences between these two main trees that give us, of course, different aspects of spatial phylogenetics. All right, and then just as a pinpoint here, just kind of interesting to sort of target one community. So you see the red dot there in the middle, and all of the species that reside in that red dot are found, are highlighted in red on each of these two trees. So you can really see like the difference in the phylogenetic diversity metrics that you might get if you're counting up, all the branch lengths of the species on the chronogram on the left for that community versus all the branch lengths for the species that are found on the phylogram from that same community right there in northern Florida. All right, so just kind of jumping into the results here. So for phylogenetic diversity, we we found a couple of interesting things. So of course we have this much higher phylogenetic diversity in central Florida if we're just looking at this metric on its own, and you do see this really interesting distinction where that third ecoregion kind of switches from central to down into the other Everglades, where phylogenetic diversity does seem to change quite a bit, both for the character divergence tree, the phylogram, and for the chronogram shown there on the right. And then if we just ask, all right, how many of these metrics are significantly higher or lower than we would expect by random chance? That's what we're showing here. So in the red dots are those where the phylogenetic diversity is less than expected by chance, is lower, pardon me, than expected by random chance. And those in black are where we see more phylogenetic diversity than expected by random chance. And I just want to point out two things here. One is that if you look on the tree on the right, or the map on the right, what we see is this really kind of interesting area in North Florida. Yeah, thanks, Israel. Well done. In North Florida, where you have, a lot of these trees that are kind of these plants that are sort of a mix of a lot of the northern species and some of the Florida species. So we think that might explain why we have significantly longer branch lengths than expected by chance in the north. And then if you look at the central part of Florida, we see a lot of phylogenetic clustering, and a lot of this, and it's found both on the character divergence and survival time. And we think this really could highlight this Lake Wells Ridge habitat, where we see potentially a lot of endemism and a lot of plants that have been adapted and isolated from the mainland for some time. So we can potentially see some of these processes that we know about highlighted on the map with these metrics. Okay, so just again, we do see in some ways these phylogenetic diversity patterns highlighting these ecoregions, particularly if we just look at phylogenetic diversity itself. We can see some stark differences between the central and the southern ecoregions shown there on both of these trees. And then if you'll go to the next one. And of course, these differences do highlight some of these different processes, which I kind of just went over. So I think we want to just jump into the protected areas. So if we show these areas that have these significantly high or significantly low phylogenetic diversity metrics, and then we ask, all right, well, what are the protected areas in Florida? So are these areas that are protected, are they those that we would pick out, right, from a spatial phylogenetic standpoint? And in some cases, yes, and in some cases, no. Like, for example, this area in the north of Florida, where we have a lot of these really, really long, aged communities, they don't tend to be kind of in some of these protected areas, where we have a huge protected area in South Florida where the Everglades are, but that may not be, highlighting some of these regions right in Central Florida where we have a lot of these endemics, and particularly, along the west coast or the east coast of Florida, where you have Miami, West Palm. I don't know if you can highlight those, Israel, but anyway, where we have a lot of these kind of really interesting, communities there. And we also have, massive development. Those might be a real primary interest for conservation. Okay, and I want to just kind of bring up two quick other pieces to kind of think about when we're doing phylogenetics, and that is scale. Of course, scale matters. We know this in almost every single study we do. And so one of the important questions we ask is, how do we choose the appropriate scale in terms of grid size? So I've just placed a grid on top of Florida, so you can see if we were to select a grid size of these squares. So of course all of these metrics are done with by each of these little dots are a square, and we've defined the size of the squares of those communities, right, that we're looking at. So if we change the grid size, you can imagine you might get very different patterns. And so that's something to really think about. Of course, the smaller the grid size you go, the more computationally intensive these analyses do get. So there's always a balance between computational abilities, powers, and resources, and grid size as well. But the other part is really just kind of thinking about what are the questions you want to ask, right? How granular do you want your study to be in terms of the processes that you might pick up? So for example, this is a paper, if you'll go to the next slide, I think it is, the next, yeah, the next facet to kind of think about, I don't know, I'm using the word facet, the next piece to think about really as sort of scale is, what is the spatial scale, right? So this scale that we chose here was based on Florida, but a few of us published this next paper, if you'll go to the next slide, looking at the plants of all of North America. And when you put all of those together, of course, you see very different patterns, right? So if you look at Florida, you really just see a lot of phylogenetic clustering, right? And you see that in fact all the way across the entire North America. So this of course tells us a little bit, which is really kind of interesting, that when you zoom all the way out, what we're really seeing is that most communities are in fact kind of phylogenetically clustered, right? But if you zoom in to Florida, you can actually see a little bit more differences in the processes that occurred to create these different habitats and communities. So thinking about the scale is really important in terms of thinking about the question and what scale you want to know. So I put a few discussion questions here. How can we use spatial phylogenetics in an informed way to help with policy? And I think we've kind of been going over that a little bit. I'm not going to jump too much into that, but how do we determine scale? And one of the scales that I didn't talk about, of course, is taxonomic scale, right? What if you just focus on angiosperms, will you get a different result? If you just focus on one clade of birds, for example, will you get a different result than if you're looking at all birds? So how do we think about those kinds of things in terms of how to design these studies to answer those questions? And then I'll just kind of take a quick look at the chat here. Oh yeah, thanks for putting the paper there. And then did you put the 2020 paper in there too, Brent?
Not yet. Unfortunately, that one's not open access.
Oh, it's not. Oh, that's a bummer. Okay. Yeah.
I'll put it up there, but I feel bad.
Okay. Yeah.
Most everything we publish is open access, but for some reason that one's not.
Yeah. Sometimes I forget what we've done and what we haven't done. Yeah. So I don't know if there's any other questions on this part. I have one final question on the discussion that will kind of lead into the next section, but I might give it a moment and see if there are any questions here before I go.
Well, on the grid cell size, I'll throw in that the kind of overriding factor there is how good your spatial data are.
Yeah.
There's hard limits if depending on how well you know the distribution of the organisms.
Yeah.
So even if we did decide we really want it to be fine scale, sometimes we can't, or we need to aim for better. So you could maybe talk about that a bit. The difference between using the actual occurrences and then doing niche modeling.
Yeah.
You get ranges that look finer scale when you do niche modeling, but they may not be accurate. So what's your thought about?
Yeah, it's such a good question. And just to sort of point out the differences between the North America study and the Florida study, that one, in the Florida one, we built full spatial distribution models. And whereas in the North American study, we were using just point estimates, right, to kind of sort this out. And with point estimates, you're limited by which points were collected, and with niche models, you have the points, some of those may be accurate, some not, but of course then you'll get these models which probably more often than not are going to overestimate the space that that your plants or other species are in. So each of these come with sort of different benefits and weaknesses, but being
Right.
aware
We sort of
of
thought that might be a good reason to do both,
yeah.
because when you do the actual occurrences, you're underestimating it for sure. And when you
Mhm.
do the niche models, you're probably overestimating it. So if
Exactly.
both of those gave the same result, then you're probably bracketed in on whatever the actual range
Yeah.
sizes are.
It'd be neat to sort of compare the two and then, yeah, and then if you could come
Well,
up with
and
like
just
a
sort of go
decent,
for robustness, like
yeah,
maybe just say if it doesn't matter, then I have a solid result.
yeah, yeah, that's exactly right. Yeah, if they match, that's quite lovely. Yeah, I think the thing with the distribution models is, you're gonna get a model that's predicted anywhere, even if, you have a building in an area, right? So being able to put, these other sort of layers on top and being able to say, all right, it's not here because this is, Miami or these other areas where it's unlikely these plants are growing. So you can actually get the actual range of a species versus the realized range, I guess it's called, is really kind of critical. But point estimates can help with that. Although you could, if they were collected in the past, you could still get point estimates, where there's a building today. And you definitely don't want to put a lot of efforts into conserving an area where there's nothing to conserve left. Great. So I have one final question, and this is something that we've been struggling a bit with, and this is going to lead into the next section, which is how can we build pipelines that allow us to scale up into larger and larger data sets? And this is something that we've spent a lot of time thinking about. We really, to do these analyses, you need massive data sets on a number of fronts. You need the spatial data, you need the molecular data, and of course you need computational resources to be able to even, do, particularly the randomizations are incredibly computationally intensive. And so, we don't, as we said, niche models can in fact over predict. And so if you're doing a project, in this project, we had about 1,500 species. So how can we, are there ways that we can actually look at each one, right? And say, we trust this model or we don't trust this model. And what happens when you go to even bigger sizes than Florida? And so with that, I'm going to introduce Tal Kinzer, who's a graduate student at the University of Florida, a good collaborator, and friend here is going to talk about part of his PhD research, which is really looking at an even larger area. Thanks, Tal.